Hello everyone. Now we would like to present on the gene expression-based classification of Alzheimer's disease. We are Chong Kah Wei A20EC0027, and Firdaus. First, I would like to introduce on the Alzheimer's disease. So Alzheimer's disease is a type of neurodegenerative disease that causes continuous and incurable brain damage with slow onset. The symptoms including cognitive impairment, behavior change, memory impairment, and the patient might lose the ability to conduct daily activities. There are many factors which can increase the risk of getting Alzheimer's disease. The first one is the genetic factor, which mainly affect the elders. Other than that, Alzheimer's disease also can happen a person's 30s to mid-60s, which is caused by genetic mutation. Next, I would like to introduce on the recursive feature elimination, the RFE. So this is the method we implemented, which it fits a model and remove the weakest feature until the specified number of feature is found. And then we use this method also uses a variety of machine learning methods to filter characteristics, identifying and then getting rid of the least significant genes among them. Here are the objectives of our project. The first objective is to apply recursive feature elimination methods for classification of Alzheimer's disease. Next, this project also is to utilize the classification models of the recursive feature elimination, and lastly, to diagnose and classify the data set of Alzheimer's disease. This is the flow chart of the proposed solution. You can see from the graph, firstly, we get data from the gene expression omnibus, which is the GEO, and then we do the data pre-processing. After we get the normalized gene expression data, we do t-test and filter less significant gene. Next, we create the model architecture. And then we split the data set into 70% of training data and then 30% of test data. Lastly, we do prediction and also evaluation on the model. Next, I would like to pass to Kah Wei to further expand on the process of our project.
All right. So I'm Chong Kah Wei, and I will continue the presentation by explaining on the data pre-processing and also the exploratory data analysis. Firstly, the series matrix file of our data set is obtained and downloaded from the GEO. It's actually a TXT file. We then copy the content of the series matrix file and paste it into the CSV file for further use. Then we delete all the unwanted data from the CSV file. We extract the information which can differentiate the state of the samples and convert it into two states only, which are the normal control and also the Alzheimer's disease state by using the formula in Excel. So at the end, the CSV file of our dataset left only the samples and also the gene expression data. So we can actually see from this figure below, we can see that it consists only the prop ID and also the samples name. While for the another dataset here, it actually consists of the samples name with the respective state. So we will actually combine both dataset together after we have done the data wrangling process. Okay, so now we look into our data wrangling process. Our original data is actually well formatted since all the data is in the flood data type and has been undergoing CGRMA normalization. Furthermore, there is no missing value in our dataset too. However, what we need to do is to convert the prop ID in the dataset to the gene name or the gene symbol, since there might exist some missing values and duplication of genes after the conversion. So to convert the prop ID, we need to obtain the annotation soft table from the GEO for our dataset. The annotation soft table is as shown in the figure. As you can see that it consists of the prop ID, the gene title, the gene symbol, and other related information. Then by using the Python, we import the CSV file of the annotation soft table. The prop ID from the Alzheimer's disease dataset is then matched with the prop ID from the annotation soft table, and the prop ID is replaced with the gene symbol. Okay, the coding and the result is shown in this figure. We can see that the probe ID is already replaced by the gene symbol over here. Alright, so after the replacement, we check on the null value on the gene symbol. So we can see from this figure, there's actually a total of 9,557 missing values, and we drop all the rows without a gene symbol. Right, then we also check on the duplicated gene. So there are a total of 22,931 duplicated gene, and we remove them by taking the average value. Alright, so at the end, the data set left only 22,187 genes after removing the missing and the duplicated gene. Okay, then we combine the data set with the stats of each samples and transpose the whole data set. So now we can see that the row become the sample name and the columns become the genes. So in this case, we actually use the sample's name as the cases and the genes as the variables. Alright, now I will explain on the exploratory data analysis. So for our exploratory data analysis, we did an independent sample t-test to find the p-value for each gene from two independent groups, which is the normal control and also the Alzheimer's disease group. So the purpose for us to do so is actually to compare the means of the gene expression value. So you can see here is our null hypothesis and our alternative hypothesis. So to perform the t-test, we use the significance level alpha equal to 0.05, which means that if the p-value of the gene is lesser or equal than 0.05, the gene can be considered as statistically significant. So basically, in this phase, we will detect the differential gene expression. Okay, so here is how we can perform the t-test. So we define the group 1 as the gene expression value of each gene for the samples of normal control, and group 2 as the gene expression value for each gene for the samples of the Alzheimer's disease. So the variance of the two groups is actually assumed as not equal since both groups is having a different number of samples. Okay, so as you can see, the equal var parameter over here is set by false. And then by using the SciPy, we can perform the t-test and get the p-value for each of the gene. So you can see here is some of the result for our gene and also the p-value. So the left part is actually the gene name, and then the right part is actually the p-value for the respective gene. Alright, then we filter the gene and left only the gene with a p-value lesser or equal to 0.05. So at the end, we left only 8,836 genes, which are considered as statistically significant and might be used to differentiate between the two groups of samples. Alright, so after we obtaining the useful genes, we filter the genes in our data set as well by dropping the columns for the less significant genes. So now we can see our data set left only 253 rows and 8,838 columns. So the columns actually consist of the sample name, the stats, and also the significant genes. Alright, so that's all from me, and now I will pass to my other teammates to continue the presentation. Thank you.
For the model development, there are going to be three main things that I'm going to mention in this part. The first one is train and test. It is to measure the performance of machine learning algorithms. The second one is random forest classifier. It is to evaluate each feature's importance and measure model's performance. And the last one is RFE. It recursively eliminating weakest features until the specified number of features is reached. For the source code, first we convert the state for each sample to number as well, whereby normal control is equal to 0 while Alzheimer's disease is equal to 1 for further analysis. The size for the train is set as 0.70 and the size for the test is 0.30. Then, the data of the train and test are saved into CSV files. The head method is used to get the first 5 rows of the trained dataset. Then, we use the shape property to get the rows and columns of the trained dataset. We proceed with printing the number of data for normal control and Alzheimer's disease in the trained dataset, where the value count for the state is 0 and 1 respectively. The same process is used for test dataset too. We create a copy of the trained and test dataset using the copy method with number as the data type of the data, then save it as xtrain and xtest. We also exclude the sample name column in both xtrain and xtest dataset by using the drop method. After that, we created ytrain and ytest to save the state column as the dataset. Next, we use recursive feature elimination (RFE) to select the important features, which in our case, the genes. We use random forest classifier as the estimator of the RFE. We built the model by using the parameter and estimator equal to 45 and random state equal to 1. For the number of features, we select the top 1000 most important features by setting the parameter and feature to select to 1000. The step parameter is set to 1, indicating that only one feature will be removed at each iteration. Using the xtrain as training data and ytrain as target variable, the fit method is used to fit the RFE object to the training data and identify the top 1000 genes from the training data. I will proceed with model evaluation. There are going to be 4 processes in this part. The first one is accuracy score. It is to find the accuracy score on both trained and test data. The second one is comparison between the actual state and predicted state. Next is confusion matrix. It is to obtain the sensitivity and specificity of the model. And the last one will be F1 and MCC methods to measure and evaluate the model's performance. We started with predicting the labels of data values of the trained model using the predict method. The X-trained data is used to be tested. Then, we proceed with finding the accuracy score for the trained dataset by using the accuracy score method. The prediction train is the predicted label, while the Y-train is the true label. We print the value of the accuracy score of the trained dataset. The same process is applied to test dataset too. Next, we create a list name as data with test data sample name set as sample name, ytest set as actual, and pretest set as the prediction of class for the column header. The data frame of the list is printed using the data frame method and assigned to dframe data and printed. This is to compare the actual ytest state and its predicted state. The different data then saved to the CSV file. We proceed with confusion matrix where the ytest is used as the actual state and pretest is used as the predicted state. Then, we obtain the sensitivity and specificity of the model after obtaining the values of confusion matrix. We ended the evaluation with using F1 score and Matthew correlation coefficient MCC.
In conclusion, after we gain the data from the GEO and then we do pre-processing, we also do t-test and also filter less significant gene. After that, we also create the model architecture and then split the data set into 70% of training data and 30% of test data. In this project, we implemented the recursive feature elimination (RFE) techniques by using random forest classifier as the supervised learning model to train the data and identify the most important 1000 genes that can be used to differentiate between the two states. To check the performance of the model, we also did several model evaluations, including the accuracy, sensitivity, specificity, F1 score, and also MCC score. In this project, we have learned more about the machine learning algorithm, such as the random forest algorithm. Furthermore, We also get idea on how computing can help in biological field. In short, this project has brought a lot of new experience to us, and we are sure that the experiences we gain from this project can be very useful in the future. That's all from us. Thank you.